Hello everyone, welcome back to Python for Bioinformatics. In the previous lesson, we learned how to count occurrences inside a DNA sequence using the count method. And today we are looking at another very useful string method called split, which helps us separate biological data into smaller pieces. Now suppose someone sends us DNA sequences in this format. So instead of 4 separate sequences, everything is stored inside one string and separated by commas. So before we can analyze these sequences, we need a way to separate them. So how can Python do that automatically? Conceptually, we have one large string and we want to break it into multiple smaller strings. And the comma acts like a boundary marker. Wherever Python sees a comma, it can split the string into separate pieces. So the method used for this is called split method. Its syntax is sequence.split separator. The separator tells Python where to cut the string. After splitting, Python returns a list containing all the pieces that were created. So here we have one string containing 4 DNA sequences separated by commas. And we have stored this one string in a variable named as DNA_data. Next we call the split method on the string. And inside the parenthesis we provide a comma because this is the character separating our sequences. So when Python runs this line, it searches for every comma and cuts the string at those locations. And we get the output as a list. Each item in a list is now an individual DNA sequence. So we started with one string and ended with 4 separate DNA sequences. Now let's do something useful with the result. Here we store the output of split method inside a variable called sequences. And then we use the len method to count how many items are present in the sequences variable. And Python returns 4. This means 4 DNA sequences were found. Notice how splitting helped us answer a biological question about our dataset. Commas are not the only separators we can use. In bioinformatics, headers and metadata often use other symbols. Here the separator is a pipe symbol. And by writing like this, Python cuts the string wherever it sees that symbol. And we can see in the result, we have gene name, species, and chromosome information as separate pieces. This makes the data much easier to process programmatically. Now this is the main biological workflow. We often receive raw combined data. Using split, we separate the information into individual elements. And once separated, each sequence or metadata field can be analyzed independently. Now let's try a quick practice question. Pause the video here and predict the output before running the code. Think carefully about where Python will split the string. Here the answer is a list containing tumor, normal, and control. Python found the commas and used them as cutting points. And each sample name became its own list element. This is exactly the same idea we used with DNA sequences. Now let's summarize. The problem was that multiple values were stored inside one string. And the solution is the split method. We provide a separator and Python returns a list containing the separated pieces. The key takeaway is that The split method transforms combined text into individual items that are easier to analyze. Thank you for watching. Today we learned how to use the split method to separate biological data stored in strings. In the next lesson, we'll learn about the join method, which does the opposite and combines multiple pieces back into a single string. See you in the next video.